Elena De Cecco from the University of Zürich, and my project today revolves about the characterization of the role of the Glycoprotein Nonmetastatic Melanoma protein B, GPNMB for friends, in Prion diseases, and its the evaluation of its potential as a therapeutic target in Prion diseases. So the idea of the project stems from the observation that so far in prion diseases, the only gene that holds potential as a therapeutic target is the gene encoding for the prion protein itself. Few other genes have been linked to the disease, but it's unclear whether they can really represent therapeutic targets, and also their role in the pathology is still a bit unclear. On the other hand, the GPNMB protein recently emerged as a key player in several neurodegenerative diseases, including Alzheimer's disease and amyotrophic lateral sclerosis. And so the GPNMB protein is a transmembrane protein with a portion that can be cleaved off and released outside of the cell. And this portion of GPNMB showed a neuroprotective effect in several models of these diseases. So our idea was to first check whether the GPNMB protein is involved in prion disease progression, and if yes, if it can be used as a potential therapeutic target as in other diseases. So now, in order to understand the approach we're using now, I'd like first to show you what we found regarding this protein, and then we will go back to the technical details. So we wanted to see whether GPNMB plays a role in prion diseases, and we performed spatial transcriptomics on brain of mice that were infected with prion diseases. And what we saw, and you can see it with these dots here, is that at a certain point of disease progression, starting around the mid-phase of the progression and then increasing over time, there is a small population that starts expressing the GPNMB protein, and this expression increases over time. So we wanted to see what this population is, which cell types are, and we identified a subset of microglia. So microglia are the cells that are responsible for cleaning up the brain from cellular debris. And we saw that the GPNMB was upregulated in this subset of patients, and the upregulation was caused by the presence of dead cells and not only of the prion deposits. And what GPNMB was doing to these cells, it was promoting their phagocytic capacity. So cells were phagocytosing more debris as if they were trying to clean up the brain area more efficiently. Then I also told you that the secreted fragment of GPNMB could play an interesting therapeutic role in disease. And what we observed is that in the cerebrospinal fluid of patients with CJD, we had an increased level of this soluble fragment of GPNMB. So we decided to see whether it was playing any role in the pathology. And we used a mouse model. Here. Where we basically collect slices of the brain, and we can infect these brain slices, with prions. And the slices can reproduce the pathology in terms of neuronal loss within a limited time frame, and we can also manipulate them and test compounds on those slides. So what we did was to infect slices from the cerebellum of mice, and we treated them with the recombinant protein that corresponds to the shedded fragment of GPNMB. And what we could observe is that the normal loss of neurons, which is represented here by a decrease in the neuronal marker nu-n, which happens when prions are present, was almost completely rescued when we were administering the recombinant fragment of GPNMB. And we also assessed that whatever was going on was not dependent on the amount of prions. So you can see here that the prion deposition remained the same while neurons were more alive. So all these data point towards the role of GPNMB in prion diseases and a potential use of its fragment as a therapeutic target. So now what we want to do is to see how the expression of GPNMB is modulated in the cells and if we can find genes that first of all promote the expression of this protein. I told you that the protein becomes upregulated at a certain stage of the disease, but if it is really helping the disease, helping protecting from the disease, then it would be useful to upregulate it a bit earlier. And second, if we can identify genes that promote the cleavage and the release of the soluble fragment of GPNMB, this could also have a positive effect on to counteract the neuronal loss. So to do that, we plan to use a technology based on CRISPR-Cas9. So you probably know that the CRISPR-Cas9 system allows us to directly manipulate the expression of target genes. We can either completely kill their expression, we can increase their expression, or we can temporarily silence them. And our idea is to tackle every gene of the human genome, trying to see what they do to GPNMB expression and shedding. So we have devised a first screening strategy where we will take a human cell line, specifically a microglial-like cell line, and we will modify each gene of the genome using our CRISPR collection of plasmids that are also called CRISPR libraries. And then we will check for the levels of GPNMB expression upon modification of each single gene. And then the genes that promote the expression of GPNMB will also be tested for their effect on shedding of GPNMB, so on its cleavage. And in this case, we have devised another strategy based on ELISA assays that are able to capture and quantify the amount of the soluble fragment that is released in the medium. So, in general, we have seen that the idea of increasing GPNMB expression and promote its shedding could be a potential therapeutic strategy. And our idea would be, once we have identified target genes that promote the expression and the shedding of GPNMB, they can also be tested with several known compounds to see whether some of these already approved drugs can actually act against our selected genes. We also have available a platform to screen new drug candidates that could act on these targets. At the moment, we stand at the step of the screen, so we have devised a cell model that faithfully replicates GPNMB expression and shedding, and we have our CRISPR libraries ready, and at this moment the screen just started, so we're currently screening for all these candidates, and we hope to find interesting therapeutic targets. Then I would like to acknowledge the people that are working with me on this project. Professor Adriano Aguzzi, who could not come today. He's the head of the lab and he's involved in prion research since more than 30 years. Dr. Davide Caredio and Giovanni Mariutti. These are my colleagues who actually performed all the experiments that I showed you. At the moment, Davide moved to another lab to go on with his career, and Giovanni is currently performing the screen. And then I would like to thank the CJD Foundation for awarding us this grant, and especially the Andy Lewis Memorial and the Jeffrey A. Smith Memorial for funding us. If you're interested in knowing more about what we found on GPNMB, I invite you to check our paper that is currently on bioRxiv and will be published soon. And here you will read all the details of the story I told you today. Thank you very much for being here, and if you have any questions, please ask me. Thank you.